Thank you so much for the introduction. I have been introduced, the topic that I'm going to talk today is about the assessment of commercial drug compounds in an engineered hard tissue platform using human-induced pluripotent stem cell-derived cardiomyocytes in a serum-free media. So I'm Manuel Rivera, and I'm part of the University of Twente as a postdoctoral researcher. But before we start, it's important to mention that the human heart is one of the main organs, and it mechanically pumps blood with nutrients and oxygen through the human body. And the cardiovascular disease, that they are the main group of disorders that affect the human heart, are the main cause of death and disability worldwide. And as you might know, the benefits are not expected to happen soon, because usually the drug development takes on average 12 years. And then from the drugs that have been developed in the market, they only benefit a small group of patients. And for the patients that they are not benefit, they also have the probability to have side effects. So what is happening right now with this? It's like during the process of drug development, we start with a big stack of possible compounds. And at the end only of the process of the 12 years, you only get with one compound that is possible to be beneficial from the human body. But what happened? During the early stage of preclinical assessment, they use models that they don't really resemble the human situation. And it's here where we see that the process to have a more efficacy and reliable drug compounds is hampered. And then what we really need to do right now is to increase the patient safety and reduce the time and cost of drug development. And for that right now, what we would like to have is an in vitro model that resembles the in vivo situation that can be used not only for drug testing, but also for cardiac disease modeling and cell therapy. The question comes now is, how close are we to achieve this goal? So in this case, it's like right now in the University of Twente, we are able to use human iPSC-derived cardiomyocytes and obtain these cardiac cells. But what is the main problem with these cardiac cells? Well, these cardiac cells resemble 16 weeks of development, so they are in a really immature phenotype. And what you really would like to use for a really model that resembles the in vivo situations are really mature cardiomyocytes, that they have the similar contractile force, a more defined shape, and they use a more fatty acid pathway of energy. And as you might have seen during these 2 days of conference, right now the 3D in vitro model have shown the advantage to resemble cell-to-cell interaction, tissue functionality, and organization. So if we can combine, as the keynote this morning mentioned, cell biology and technology, we can get closer to an in vitro model that can resemble the in vivo situation, and we can use it in this application for drug testing. And then for that, we have right now in-house a platform that is versatile and robust, that fits into a commercial 12-well plate, and is a PMMA base. And with this platform, we can make 3D cardiac tissues, as you can see here. These 3D cardiac tissues are formed around 2 flexible pillars, and then by having it per well, we can make three tissues per well, only using 250,000 cells, which allow us in this format to have 36 tissues at the end. And as you can see here, then we are able to fabricate these 3D cardiac tissues. But this is not the only important value that we have. It's like, of course, you need to quantify what is the contractile output if we want to compare what will happen in the in vivo situation. So what we do is like we follow over time the position of these flexible pillars from the relaxed position to the contract position, and then by taking into account the position of the tissue, the geometry, and the mechanical properties of the material, we can quantify the contractile properties. And we don't only quantify the amplitude of the contraction, but also we quantify the contraction kinetics, which means the velocity of contraction and relaxation and the time that takes to go up and down in terms of 10% and 90%. So this is really crucial because you would like not only to know how it's affecting the contractility when you apply different drug compounds, but also how it's affecting another parts of the cycle of contractility. And so far, I have been showing you just animations and designs, but you will say, okay, how easy is this platform that you are just presenting to us? So in this short video of 30 seconds, you will see how easy it is to make these 3D cardiac tissues in our platform. So as you can see here, in the 50 ml tube, we have the cell mix, and then you just need to pipe it inside of each one of the wells 3 times to make the 3D cardiac tissues. And after pipetting inside of the wells, we have the shapes of the tissue. You just need to put on top the holders that come with the flexible pillars, as you can see here. So, of course, this video has been speeded up just to the keeping within the time frame that I need to present. But I can tell you that in the group, of course, we have the PhD and the technician that they are really able to make the 36 tissues really fast in the lab. And this is just a quick look how it looks in real life. Now we move towards the main point. So can we use this platform to do drug testing? So how it works, the experimental pipeline that we were working on it is like first in-house we are able to use iPSC that also can be patient-specific. We differentiate it to cardiomyocytes, and also we have a step where we do lactate purification to obtain the higher percentage of cardiomyocytes before we freeze them. And this is an advantage because then we are not dependent of how the differentiations are going per week before we just establish the experiment. So just we have a big batch of cardiomyocytes, and then when you have everything prepared, you can run the experiment. And then when we make the tissues, what we additionally add is to have a more structural tissue is fibroblasts. So we add 3% of fibroblasts plus the cardiomyocytes. And then after making the tissues, we keep the tissues alive for 10 days, and we have previously shown that the higher contractile output of these 3D cardiac tissues is after 10 days of tissue formation. And then at day 10, keeping in humidity control and temperature, we just do the drug assessment. And in the drug assessment, we first have a baseline measurement when we have electrical stimulation. And from there, we added, depending on the condition, the positive inotrope, the negative inotrope of the non-effect compounds. So we wanted to see if these 3D cardiac tissues were allowed to show what has shown in literature that, okay, they improve the contractile output, they completely decrease the contractile output, or there is no effect in these 3D cardiac tissues. And then, as a result, as you have seen here, and I just showed you in the previous slide, we can have contraction force, we can have contraction velocity and relaxation and time. But due to the time constraint, I'm just going to focus right now in force of contraction and contraction velocity. So what we saw in the case when we were working with the positive inotrope is like the positive inotrope and the negative inotrope. So in this case, in green you will see the positive inotrope, and in red you will see the negative inotropes. Is like in the case of the positive inotropes, the one that they are supposed to increase the contractile output when the tissues are under this stimulation, is like in 17% of the cases we got the expected drug response. And besides that we got the drug response, in these 3D cardiac tissues we saw that they were higher sensible to small concentrations. And the one that they were not within the range, that in this case was just only for digoxin, was around or highly above what is expected in literature. And then from the negative ones, what we saw that we were able to got an expected result of 87% of the negative controls, the negative inotropes. And in this case, what is interesting to mention is like only in one of the cases we were able to see an IC50 above the usual expected results. And this is just the first parameter that you usually look in terms of contractility. But then comes the part, okay, we want to see how it's affecting also the contraction velocity and the relaxation velocity. Before we move to that one, if you remember, I mentioned, okay, we have the one that increased and the one that decreased, but let's see what happened with the compounds that they are supposed to don't create any effect on the contractility. And we were able to detect that in the case of enalapril and aspirin, that was not effect. So this is really good because this is what we were expecting. And coming back to the contraction and relaxation velocity, so in the case of the positive inotropes, what we clearly saw is like that the relaxation velocity was increasing between 10 and 20% in all of the cases, correlating with the increasing contractile force that we saw previously. And then in the case of the negative inotropes, what was really clear is like in these specific cases, after 1 micromolar concentration, then we were seeing a clear decrease in the contractility. Trying all of these compounds then give us the result to say, okay, what is what we are seeing with this platform and what is the main message with this platform? Is first that we have a versatile and robust platform that allows us to make 3D cardiac tissues, and that we can use human iPSC, and in some cases, depending, we can also use patient-specific cells. And that we not only are able to make the 3D cardiac tissues, but we use serum-free media, which avoid the protein bonding, and besides that, resemble more physiological what we have currently. And in general and overall, we were able to detect in 80% of the cases the drug response that is in literature. And the main message is here that we have a human-based in vitro model that can be used during the early preclinical phases to evaluate what is the possible response when you want to use specific new developed molecules for the drug applications. And before I finish, I would like to acknowledge the two groups that I'm part of, the BIOS Lab on a Chip group and Applied STSM Technologies, and the Netherlands Organs on a Chip initiative that I'm a part of. And with this, I will be happy to answer any questions.
Thank you for the presentation of your model and the specific results. May have one. So did you compare compounds which have a high protein binding in the system with and without serum? Did you see a difference?
No, in this case we didn't compare. What we did was in a previous result we were just comparing what happened when you use serum in our media and what happened when we use serum-free in our media in terms of how they improve the maturation of the cardiomyocytes. And in that study we managed to see that when we use our media in-house without serum-free, we get similar results or even better results when you use serum. So that's the reason we didn't try it in this one to see the binding.
Okay.
Very nice talk. Very cool to see the entire overview of your PhD like this. I was wondering, for the compounds that you couldn't predict the proper effect, do you have any explanation as to why that is? The lack of expression of the specific protein being targeted or something else?
Well, thank you so much, Ruben, for the question. I think that there are many factors that we could just try to guess why we were not getting the response. One could be that the compound works in a higher concentration than the concentration that we were working on. And another fact is, like, as the Professor Gordon was mentioned early this morning, is like you are multiple stages of maturations. And in this case, we need to work towards more electrical stimulation to have a more mature 3D cardiac tissue where we can say, okay, maybe those compounds really require a more mature cardiomyocytes.
Hi, thanks for your presentation. I really like it. Have more like two technical questions. Do you use the MCO to resuspend the compounds you've tested?
No, we didn't test that compound.
And how easy is to measure all the contractile dynamics? Like you do it with the microscope, but then all the data analysis, it's kind of automatized, or you have a poor PhD student then? Like
Okay, thank you so much for the question. So then regarding how is the analysis done, right now we have an automatic system to analyze the contractile output because I was the poor PhD at the beginning doing the analysis. So I identified that indeed, if you want to transfer the technology and make it more accessible, you really need to make it easier for the people that is in the lab using it. So right now we have the full pipeline when you just make the tissues as easy as you saw it, and then later on you just load it in the software and you get all the data.
Thank you.
Maybe one more question. I don't see any. So thank you again.
Thank you.
For your presentation.